Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Combination Drug Products: What Lies Beneath? Risk Analysis for Product Development. My name is Sydney, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Experic, who developed the content for this presentation. Experic, a contract manufacturing organization and pharmaceutical supply services company, supports every phase of a product's lifecycle from clinical to commercial scale across a range of dosing and packaging formats, including capsule filling, powder and pellet dosing, including DPI, and auto-injectors and pen assemblies. Utilizing cutting-edge Harrow Hofliger equipment in its state-of-the-art Class A GMP facility and built-to-suit suites, Experic manages global delivery of the highest quality products, even for expedited projects, while providing unparalleled knowledge, expertise, and customer service. And now I'd like to introduce our speakers for today's event. Justin Lacombe is Chief Scientific Officer at Experic. Justin leads the pharmaceutical development and engineering teams to provide expertise, insights, and practical solutions that address many of the technical challenges that pharmaceutical companies encounter during drug and combination product development. Justin's focus is on robust scale-up and process development using quality by design and design controls principles. He has specialized expertise in manufacturing, combining small volume powder dosing technology for rheologically complex powders, primary packaging development, and formulation optimization. Justin earned his Master of Science and PhD in Chemical Engineering from Rutgers University and has presented extensively on topics of manufacturing drug-product interaction, process development optimization, and drug-device combination products. Dr. Julie Suman is co-founder and president of NextBreath, an Aptar pharma company, a contract research organization dedicated to the development and analytical testing of nasal, inhalation, and injectable delivery systems. She also supports scientific affairs for Aptar Pharma. Dr. Suman directs the research division that supports product development and regulatory submissions for North American and international clients in the pharmaceutical, biotechnology, and medical device markets. Dr. Suman holds a Bachelor of Science in Pharmacy from Duquesne University and a PhD in Pharmaceutical Science from the University of Maryland, Baltimore. She's a co-editor for Respiratory Drug Delivery Proceedings, an international symposium, and an adjunct assistant professor at the University of Maryland School of Pharmacy. She's also an affiliate assistant professor in the Department of Pharmaceutics, School of Pharmacy, Virginia Commonwealth University. Dr. Suman is past chair of the AAPS Inhalation Technology Focus Group. And now, without further ado, I'd like to pass the presentation over to our first speaker. Julie, you may begin when ready.
Thank you very much for the kind introduction. And Justin and I from Experic are extremely happy to share this presentation with you today related to combination drug products. And we're going to go into what I hope is a very meaningful webinar and an expansion of a webinar that we did earlier this summer on combination drug products. So our scope for today is to talk about the guidelines as they relate to combination drug products and that regulatory landscape that is out there. And in doing so, we hope to highlight the concepts underlying the interplay between quality by design and design controls, the critical quality attributes that relate to inhalation drug products, and risk management. And we're going to do that in the context of a case study using dry powder inhalers as the example. So when we start talking about combination drug products, our sponsors or the pharma companies, we're all used to following drug GMPs as it's defined in 21 CFR Part 211. However, it has been well established that dry powder inhalers, which is the DPI, meter dose inhalers or MDIs, and nasal sprays are combination drug products, which I'll abbreviate as CP throughout this presentation. And in fact, there was an FDA guidance for DPIs and MDIs issued in 2018 that specifically points out that these products are combination drug products. Now, there are also GMPs that relate to combination drug products. Those are defined in 21 CFR Part 4. So, this poses a question, as a pharmaceutical sponsor of a drug product, what GMPs do you follow? So the way to approach this is to utilize, our standard or traditional GMPs and add in 4 components of the medical device aspects for GMPs that are defined in 21 CFR Part 820. So this streamlined approach of combining our traditional drug-based GMPs with medical device GMPs for combination drug products includes the addition of these 4 pillars, which you can see here on the screen. It starts with management responsibility. And this is really defined as having someone with a senior or an executive level type of oversight in the entire process of the product development through validation. So it really starts with being engaged at the very beginning when those user needs are being defined. Obviously, a very key component of any quality system is a CAPA or corrective and preventive action system that is also very well defined in these pillars that you see here. Purchasing controls is also defined or described in this new section. Probably all of us do have purchasing controls in place, but wanted to highlight one aspect of this that is specifically called out, and that's the need to have a quality agreement between the supplier and the sponsor. And that quality agreement really should have a very well-defined change controls section as well. And finally, there's design controls, and we're going to spend a lot of today's webinar talking about design controls and how they relate to quality by design. So we're going to walk through some of that in these subsequent slides. But first, I want to talk about how the device relates to the combination drug product. And I'm going to walk you through this slide. So if we're going to start at the bottom, so many of you are probably sourcing your device components from a supplier or multiple suppliers. They are considered critical components. And in the inhalation landscape, they could be the valve or the actuator for the meter dose inhaler, the actuator for a nasal spray, or the DPI engine for your dry powder inhaler. Those components are recognized as the device constituent parts. And when the sponsor combines them with the secondary packaging, whatever that may be, we now have a container closure system. So it's the device plus the packaging. We combine that with, or that is combined with the drug constituent part or the formulation. And when you marry those things together, the device, the secondary packaging, and the drug, you have a combination product. You'll see here on the far right-hand side that there is a potential gap between the supplier and the sponsor. It's pretty obvious, I think, from the slide that there are aspects related to the device that become a part of the information that's gathered into that combination drug product overall design history and filing. And this is where your device supplier can really help fill some of those gaps. Now, if we take a step back and look at the process from a quality by design standpoint, part of our GMPs that are also defined in ICH Q8, which talks about the design, quality by design process. The meter dose inhaler, dry powder inhaler guidelines that I talked about in that was released in 2018, while this is a draft guidance, it still does reference this ICH QBD type of concept. And just to kind of recap on what QBD means. So, we're starting with that quality target product profile. You'll see it abbreviated as QTPP. These are really the patient requirements. What do we need to have a safe and effective and reliable product? That leads us to start to develop the critical quality attributes, or the CQAs. And these are specific to the drug product. And in the inhalation world, that could mean the delivered dose or the aerodynamic particle size distribution, just for example. We move into that process development phase, and there's aspects there that also need definition. Your critical processing parameters, things like the manufacturing steps, whether that's mechanization or blending, the filling processes, any sort of secondary packaging, as well as the raw materials that we are introducing into that manufacturing process, and understanding those parameters of those materials, things like density, the purity, the flow properties. And that ultimately results in having a control strategy where we are monitoring, all of these aspects. And it's really, important to understand this is a continuous process. It doesn't just start with one loop. We're really evaluating whether our design inputs or our attributes rather are really, doing what we intended them to do. And that's where design control from the medical device guidelines starts to come into play, and that we have this process in place where we're really trying to confirm that there's really no negative interactions with the combination drug product, and that we do have a safe and reliable dosage form. So it's achieved through this process that's just shown here on this waterfall diagram. Comes back to identifying those user needs. And that's the key pillar. And then it goes through a various cycle of where we are understanding the design input. That's where we're really developing that quality target profile, where we're looking at the performance characteristics of the product. There's this process through which we will determine the design output, and that's really where are we meeting our CQAs, things like our specifications, our engineering drawings, and that's all done through a verification process where we're making sure that essentially our inputs are meeting the desired outputs. We're answering the question, did we design this product correctly? Now, in this gray shaded area, there is a device component, and my parent company is Aptar Pharma. And this is where Aptar or another device supplier or your suppliers that you're working with will be involved in this process. After the verification phase, we're going to have a finished combination product, but we're not necessarily done there. There is a validation aspect to this, and that validation is really proven through our clinical trials and human factor studies to really come back to whether or not we did design the product we intended. The other thing that I want to point out on here is that there is a review process that goes on through all cycles or all portions of these phases. That's a very important aspect. And with that, I'm going to hand the presentation over to Justin to further highlight some of the points between design control and QBD.
Thank you, Julie. Regarding the differences between design controls and QBD, there's some interesting aspects that just wanted to point out as far as it's concerned. In general, there are actually a similar number of steps for QBD processes and design controls, essentially a 7-step process. But if you were to lay them out next to each other, they don't really map to the same sort of to the same places within the development process. QBD is essentially front-loaded in terms of identification of the different risks that are involved in it, and a lot of the different processes really focus around around determination or what are the critical components of the process. And of course, pharmaceutical development side, of course, does focus a great deal on process versus the design side, which is the focus of the medical device side. Regarding that, one of the things that in the QBD process that is not specifically called out is essentially the design validation step on the pharmaceutical side. And of course, Julie talked about that perfectly in the last slide. We're saying that it's essentially the clinical trials provide this validation process where you essentially have design verification, which is essentially the in vitro portion of this. Did we meet our critical quality attributes? And then those critical quality attributes have to then translate into an actual efficacious dose. And so this of course, this structure here, which I think I presented a few years ago at another symposium, this gives you an idea that there are some differences in terms of how these two interact and how pharmaceuticals tends to think of this. And the other important thing, of course, is that risk management must take place throughout the process. We say at least from the design process, and it has aspects where it can actually provide factors in the input, and then of course design human factors as well. Next slide, please. Would want to take a little bit briefly and talk about the risk management aspects of this as well. Risk management is laid out largely in ICH Q9, though there are other locations for this as well, in which when you talk about risk management, there essentially are three aspects to this, and they're all largely iterative processes. You have a risk assessment process, risk control process, and then a risk review. And at any point in time, the risks can you can iterate between either review and control, and also for controls that are not acceptable, you can go back to the assessment process as well. With respect to this, there are a number of different reasons to do risk management. Of course, we find it important to do it for the respect to the risk to the patients, and that of course is ultimately what risk management is supposed to do, is to reduce risk to patients and users if you use the context on the medical device side of the fence. But it can also provide a very good ordering for the possibilities of creating different experimentation. So largely, if you do not understand where you're starting out in your process, this can give you key insights into how to prioritize your experiments. And there are a number of different tools that can be used for doing this, of course. And I've just shown a few of these here. So we have anything from fishbone or Ishikawa diagrams to cause and effects analyses and fault tree analyses. And of course, everyone knows and talks about failure modes and effects analysis. And important in terms of the process to pick tools that are appropriate for the level of development that you're doing in terms of this. And usually that will go from more simplistic approaches at the beginning to more complex approaches throughout the process. So and the other thing, of course, I want to mention is that when we talk about it, especially on the medical device side, then there will be a number of different levels of requirements that will be necessary. We may have user level risks that are assessed, design risks that are assessed, and of course, process risks that are assessed as well. Next slide, please. So in discussion of human factors and how does that relate to design controls, human factors, of course, translates largely into user requirements. So user requirements feed in iteratively into the user needs, into the design inputs, which then feed into design inputs, which feed into design, and ultimately into design outputs as well. And of course, that's also overlaid with risk analysis. And risk analysis can also highlight, as we talked about, additional inputs that need to be mitigated, that can then also be considered in terms of the design. As far as the process is concerned, largely what I've seen is that during the clinical trial phase of manufacturing, or clinical phase, we will focus mostly on the design inputs to design outputs and verification. While the human factor studies will be going on, informative studies will be going on, the DV phase of verification for the medical device will be sort of the focus in terms of this. Later on in the process, especially when you get towards either your pivotals for phase 3 or pre-commercial, then you will be talking about your summative studies, which is your validation. And your validation, of course, as Julian mentioned earlier, is really the connection here between the initial user needs, the additional user requirements, and the medical device itself. It's not simply the numbers that go along with this. So in addition to doing our trials that show that the drug product is efficacious, we also are showing that it is being used correctly. Next slide, please. And in terms of human factors, of course, there are a number of things that need to be considered. There's, of course, the user that has to be considered, and the user, of course, could be this could have a number of different aspects to it. But we can be talking about adults, we can be talking about children, we can be talking about people with different levels of capability with respect to this as well. And it also depends on who's doing the administration with respect to it. Is it being administered by the user, by the patient? Is there an assist in terms of the user? Is this done by a physician or clinician? And then, of course, this also leads to environment. Is this, of course, going to be administered in the home? Is this going to be administered in the clinic? Is this going to be administered in the ER? So those all relate to the design of the human factors in terms of the medical device itself. And of course, there's also the specifics of how the device is designed itself and how it interacts with the human being. Beyond that, I would also like to say that you have to think about this not just in terms of what's happening ultimately for the commercial product, but the human factors can also become important for what happens in the clinic, because if the clinicians are going to use this, then this has certain implications in terms of the environmental conditions, whether or not this is a take-home study or whether this is something that is only going to be administered in the clinic, can all have impacts on the human factors that you consider during the trial phases of your development as well. Lastly, I wanted to just sort of talk about the generic space in terms of how this can work when you're talking about combination products, especially about inhalation products. There's this interest in AB-rated generics in terms of how those could potentially be used. And there are some thoughts and some guidances out there from the FDA as far as threshold analysis, as far as using studies, basically risk analysis to understand whether or not human factors are done, so that human factor studies don't necessarily have to be repeated. They talk about a threshold analysis where labeling comparisons need to be done, in addition task analysis, and then also physical comparisons can be done, and how to handle any kinds of risks that are bubbled to the surface in terms of those types of interactions. With that, I'd like to move on. Thank you. With that, we'd like to move on to a case study and talk about DPIs, and of course, relevant to combination products, because of course it involves drug product and any number of different potential incarnations or devices, whether they be single dose devices or multi-dose devices in the co-packaging aspect. So motivations in terms of inhalation, of course, is you start off with both the formulation and device. We're showing here solutions of suspensions. Because we're talking about DPI formulations, largely we're also talking about powders as well. These then interact with the device to create a specific particle size distribution, and based on that particle size distribution, typically we call an aerodynamic particle size distribution versus a geometric particle size distribution. Those could be distributed to different parts of the body, whether it be the lungs or the nasal region, if we're talking about a nasal device. And of course, it's not just about delivery of the material to the site of action. It also then has to interact with the body, and so that of course involves both dissolution and permeation in the system so that you get some systemic interaction. Though in a lot of the inhalation compounds that we work with, while there is systemic absorption, a lot of it is taking place at the site of action as well, i.e. the lungs. Next slide, please. Just a brief, contextually, we did discuss a little bit earlier quality by design and how that interacts with, how that is important to the drug development process. And of course, there are some standard pharmaceutical processes that are important for DPI development: assay purity, moisture, bioburden, to name a few. It of course is very much dependent upon your quality technical product profile and what it says. But there are some that are very specific to inhalation and nasal devices. And solubility and permeability, of course, are very important. And when we talk about solubility and permeability in this context versus the context of like an oral solid, then we are talking about more about dissolution into lung fluid or dissolution into the surfactants within the nasal cavities as well. So there are some differences in terms of that as well. And then of course aerosol performance. And aerosol performance is important for both DPIs and also for other types of nasal products as well. So while we're trying to deliver it to the lung in this case, there are cases where if you're delivering it to the nose, you want to make sure that your particle size is large enough that it doesn't actually get delivered into the lung as well. So studies are done along that lines as well. There are of course other things that are important in it. Those are dependent upon what type of device you're using or type of drug product you're working with. Unit dose uniformity, if you have pre-metered measured doses versus net content uniformity, which is important with respect to reservoir-based systems. MDIs fall into that category as well. Spray powder and plume geometry don't really apply to DPI, but are important for other types of drug products as well. Next, please. And just a little brief consideration in terms of how drug device interactions can work. Of course, when we talked about the quality product profile all the way down to critical process parameters and critical material attributes, there's of course just this aspect where they have to have some interaction with the drug device, and some of these interact with the drug device design as well. So some that don't, obvious cases like assay related substance moisture, but then of course delivered dose uniformity and fine particle fraction are very important to the drug device combination. In that respect, taking as an example an RSO1 device here, needles pierce the capsule on the side. The needles have a very specific design, so the holes have a very specific design, as well as the specifics around the capsule itself. And if those do not interact optimally or they're not designed together well, then either the drug does not get dispersed properly, and you have too much residual drug within, so therefore your delivered dose is improper, or you don't get the right particle size distribution, so it doesn't get distributed to the right location. There's a lot of different interactions here. The mesh, of course, is important as well. And then also, just as an aside, mechanism in terms of how the device works, of course, is very critical. So it's not just the design of the say the pins or in that. If you're talking about an RSO1 device versus say a turbo haler, they're going to have different mechanisms of action, and so the formulations that you're working with won't necessarily be designed in the same way. And I'll turn it back over to Julie now for the next.
Thanks, Justin. So Justin so nicely, introduced the idea of solubility and permeability, and it has a unique relationship for inhalation drug products. We're probably used to thinking these in the context of oral solid dosage forms, and so many of you on the call here are probably familiar with the BCS, the bioclassification system, that talk about how to characterize a drug in terms of its permeability and solubility, and then that may relate to how you would do various studies, dissolution studies, etc. This system, this concept, is also making its way into the inhalation world, particularly with those poorly soluble drugs. things like corticosteroids, which are very commonly delivered via inhalation. So these, the corticosteroids, for example, don't dissolve readily. And so it becomes an interesting aspect to understanding an inhalation product. So if we look on the right-hand side here, we have at the top, the apparatus is called a next generation impactor or a cascade impactor. And this is the device that we would use to determine aerodynamic particle size. You've heard that mentioned a number of times, that APSD. So this is the actual apparatus that we would use to determine that. Now, within that distribution, so each of those little stages and cups there, if you will, you'll have a breakdown of different particle sizes of your drug product. And in those various distributions, we're going to have pure drug product, or we're going to have maybe some agglomerations of the drug product with the carrier, because most dry powder inhalers are going to use a carrier such as lactose to facilitate the filling of the device. So, when we start to think about how that drug may interact at the lung surface, this is where dissolution starts to play a role in our understanding of that. And this is just an example of using the deposition from one of those stages you can see here in the picture, and inserting them into a dissolution apparatus. And this is a standard USP type system. There are other apparatus or other mechanisms that are in develop. The one, as an aside here, one challenge to this is there is not a standard way to do a dissolution test for an inhalation product. Nonetheless, it can give us some interesting information that tells us about the formulation and perhaps even some of the manufacturing processes. And I'll try to tie it together in this slide. And this data is actually from NanoPharm, and it's talking about dissolution from 3 Advair products. Advair is a dual drug combination drug product. The drugs are fluticasone propionate, that corticosteroid, and salmeterol xenofovate. It's the salmeterol is abbreviated as XX in the slide, and FP is the fluticasone. There's also lactose in the formulation. So if we start on the right-hand side, this is the dissolution profile for the 3 dose strengths of Advair. So they increase in the dose of fluticasone, but the salmeterol dose stays the same. So the red line in that dissolution profile is the low dose strength of Advair. And you can see that as you increase the dose strength, the dissolution rate is lower. So there's an inverse relationship between the dose and the dissolution rate. Now, that obviously can have effect at what happens locally in the lung and also systemically, even though we don't really want systemic absorption. But if we try to understand why that is happening, and we take a step back and look at the formulation microstructure, or Q3 equivalence is another common word that you hear here. So this middle slide is a visualization of the particulates in the dry powder formulation. So the top bar is the high strength and the bottom is the low dose strength fluticasone. Blue is fluticasone. Orange is a fluticasone salmeterol lactose agglomerate or aggregate. The yellow is fluticasone and salmeterol, and then the green is fluticasone and lactose. So you can just see here visually by the color distribution that we have a different makeup of the combination of how fluticasone is present in the formulation depending on the dose strength. Since fluticasone is poorly soluble, adding a water-soluble component such as lactose or salmeterol can help facilitate the absorption of the fluticasone or the poorly soluble product. And so if you can see in the bottom of that graph, we have a large presence of lactose and salmeterol agglomerates, and that is actually helping the fluticasone dissolve faster. So that explains the dissolution profile on the left-hand side. We could take it even a step further through various type of modeling and do some predictive pharmacokinetics. So on the far right-hand side here, we have fluticasone propionate in the plasma as a function of time. And these are different models where the dissolution rate constant is increasing. So you can see how this ties together to understanding your formulation, and even the manufacturing processes can influence, the extent of these agglomerates. How the very basic information can ultimately influence the pharmacokinetic profile. Switching to talking about aerosol performance, it is one of those metrics that we would be, following as part of our QBD process. And so cascade impaction or aerodynamic particle size is typically done by a cascade impactor shown in the upper right-hand corner with an induction port. So that is that little throat-like apparatus that's attached to the impactor. It's where we introduce the inhaler into the apparatus. Now, the standard USP testing utilizes that throat, the USP throat that's kind of got the yellow picture there. It has that 90-degree bend. It's the standard for doing any sort of QC testing for an inhalation product. But there's also a school of thought where it might be useful to utilize a realistic model or a realistic throat. Now, there are many different types of these models out here. I just have shown one, which is the Alberta Idealized Throat, or you'll see it often abbreviated as the AIT. That's shown in the red here. You can kind of see from the picture that it does have a little bit more of an anatomical design compared to the USP throat. If we pop down to the diagrams here, the left is the cascade impaction profile, the drug deposited on each of those stages that we talked about for the USP as a function of the emitted dose. And then on the right-hand side, we have the AIT throat. And, the 4 bars or the 4 lines that are shown here is actually from a generic product development program where the solid line is the RLD and the other lines were attempts to find an equivalent formulation to take forward for a bioequivalent study. And overall, you can see that the distributions are fairly similar, with the exception of the dotted lines that has where the target aerosol performance parameter, the MMAD, was larger than the RLD. You can see that it does perform differently. But when you look at the two throats, the two profiles, that you don't see a lot of differences. However, in the world of inhalation, we will have mouth throat deposition in the human, and that's what these idealized or USP throats are meant to try to identify. So here's where we can start to see some differences. So let's say you have set a target throat deposition based on the RLD here. You can see the target in the dashed lines. And the other, with the light gray bar, is the RLD. The other bars are those proposed formulations. So you can see if you're using the USP apparatus, the micronized, that light blue formulation does not meet that target profile. But if you use a realistic throat or something that is more meant to be anatomically correct, you see that you get a different answer. That micronized formulation is actually below your target. So the reason I wanted to highlight these two different types of testing procedures is that it goes back to understanding what you're trying to achieve, what your targets are, what's the right methodology to help you understand the performance of your product. And the real world kind of ramifications is that, in the patient, if you have more throat or mouth deposition, that means it's not being delivered to the lung. So that might mean you might see that have an effect in less of a delivered dose to the lung, or not meeting, your desired clinical outcomes. So again, just to highlight that there are different ways of trying to understand your aerosol performance. And we've talked a lot about the concept of particle size and small particles, and they certainly are very important in the world of inhalation. However, there's other attributes of an inhalation drug product that we also need to understand as part of our development process, and it also could potentially impact how we may manufacture it. Particle density can influence how far a particle will deposit in the lung. Shape and aspect ratios are also important in terms of how it forms agglomerates, or maybe even like a more rod-shaped particle may deposit more peripherally in the lung compared to another shaped particle. Certainly moisture and hydroscopicity play a role. Powders don't really like moisture, and if you have moisture in your formulation, it may not be aerosolizable. So it's definitely, something to understand. And then in the context of talking about the solution, surface area and whether or not you have a crystalline or amorphous product also play a role in understanding the overall underlying performance of an inhalation product. And Justin touched on this a few slides ago to a certain extent, and that if we talk about dry powder inhalers, the device or the container, if you will, plays a role in how the aerosol or the powder is developed and inhaled. And there are three sort of main kinds of dose engines, if you will, for a dry powder inhaler. There's a blister-based device. Advair is an example of a blister-based. There are reservoir devices where the drug is, sort of packaged in kind of a puck, or literally like a reservoir within the device. A turbohaler is an example of that. And there are also capsule-based devices. Spiriva is an example of a commercial product that uses a capsule. And these types of devices, they all play a role in understanding the overall performance. They have different materials of construction. They may interact with the formulation in different ways. Certainly in the context of a blister or reservoir system, the drug is in direct contact with the device components, so there could potentially be an extractable and a leachable, more so a leachable aspect that needs to be understood in those types of systems. And then just to kind of highlight on another thing to consider is related to capsules, and just to kind of further develop some of the concepts that Justin described earlier, and that, capsules, capsule-based DPIs have an interaction between the capsule and the device, and the capsule can also, be part of that aerosolization process. And shown in the picture in the bottom on the right-hand corner are different types of piercings that were done with capsules that were, exposed to different conditions. So you can see that, in some cases the capsule was shattering. And so depending on the material of construction, what it's made of, the exposure of that capsule to moisture, the design of the device, the pins themselves, and how it pierces that, all of those, those things that seem so small actually could play a major role in affecting the ability of the device to deliver the full dose as well as the right particle size. So it, just highlights this slide is, to really also make sure that, in the world of inhalation, the container closure system also plays a major role in product performance. And certainly there's other things to consider, and I'll hand that over to Justin to talk about some of the processing parameters that we need to think about.
Thanks, Julie. As Julie spoken about, there are a lot of things to consider with respect to the design and development of a DPI drug product. And of course, manufacturing and formulation is obviously of critical importance when it comes to this. For DPIs, though there are some different types of formulation technologies that are possible, the principal ones that have been used in the past are blending type formulations and then blended type formulations, typically blended lactose, and then spray dried formulations as well. Though the components of these formulations have a tendency to be somewhat different from each other. In terms of the blended formulations, typically this is done by high shear blending using a micronized milled API, so some API that is a pretty small size, and then a what we call an inhalation grade carrier of lactose typically, which is I would say principally a little bit different in terms of this the size characteristics that you would expect to see in an oral lactose blend or material. As far as what's important to this, beyond the of course the materials that you're using, which are of course important, is to understand the type of blending, is to understand of course the importance of the materials themselves, or the different interactions you can have within the device, within the manufacturing process. So when we're talking principally here, one of the key components is of course the amount of kinetic energy that we are actually imparting to the system. And it's really a balance, right, in that sense. The problem with, of course, these types of formulations is you have a carrier and you have an API, and really those two can need to be separated during the process in which they come out of the DPI, but they need to actually be well associated together while they are within the formulation. Otherwise, it cannot be consistently manufactured, i.e., doses will have different concentrations of API, which of course is anathema to what we're trying to do, which is consistency within our device. And of course, the other things that can be important here is the order of addition in terms of the components, because you can have things preferentially stick to the sides of the blender, and then of course capacity and concentration issues, and the amount of time that it is blended, and all of these interact together to create the formulation, and they can all have impacts in terms of the behavior of the formulation, both from we'll say an assay related aspect, but it can also, of course, interact with respect to the performance of the drug product itself, which is really the critical thing that we look at here. On the spray dried side of on the spray dried side, of course, then spray drying configuration is important. Of course, important. The rate in which you're drying, you're creating a set of droplets with a certain concentration within them, and those need to be dried. Depending on how the rate in which you dry, you can get dense particles, you can get hollow particles, and those particles can deform in certain ways. You can have essentially what looks like sort of bursted particles, or things that have a lot of different amount of surface area. The other thing that's important here is that spray dried materials you can also create, because you can create hollow particles, you can actually have particles that are larger in size but have lower densities, because they have lower densities are still inhalable, because inhalation is not just a factor of the geometry, it's also a factor of the density of the particles themselves as well, and those interact as well. And then next slide, please. Talking about filling, of course, filling is an important aspect in terms of getting the appropriate amount of drug delivered to any individual unit, especially when we talk about multi-dose units or capsule filling. Some of the issues that come about because we are working with these kinds of products is, we have small particle sizes, which create, which in small particle sizes have a tendency to create powders that are very cohesive in nature, which of course impacts the powder handling, and of course that impacts the consistency of filling. On the spray dried side, you may also have hygroscopic powders, which of course require you to use low humidity environments, which can create static. That again can create issues when it comes to powder handling and then consistency as well. Small dose sizes, that's more along the range of the smaller the dose size, then in order to get a consistent product, if we're trying to stay within, say, plus or minus 7% in terms of our target, or for assay usually it's plus or minus 10%, you need to then have a smaller range of acceptable doses. So if your dose is 1 milligram versus a dose is 10 milligrams versus 100 milligrams, then the acceptable range and not the relative size, but the actual size of what's acceptable in terms of a plus or minus percentage range is different. So the smaller the dose size, then the harder it is to create those kinds of consistent doses. And of course, if you have inconsistent formulations, then you're going to have inconsistent filling. and you can't expect to have consistent dose uniformity in those kinds of cases as well. There are a number of other different ways that these things can interact as well, but at least on the filling aspects, your delivered dose uniformity is really very much contingent upon your ability to fill these things properly. Next slide, please. And then just a little musing on dose variability. When we talk about what's acceptable from a delivered dose range for these drug products, there's a plus or minus 20 to plus or minus 25%, and in case the European authorities up to plus or minus 30% for the acceptable range for individual delivered doses. The thing to remember here is that that is limits that is not based on that's not an RSD. So in order to meet those targets, your RSD of course has to be significantly smaller than that, because your standard deviations, of course, you can have up to 99% of the materials will fall within plus or minus 3 standard deviations, but only 67 fall within 1 standard deviation of the mean. So in those kinds of cases, you have to consider that your overall variability, really, if you're talking about 30%, is really only, if you're expecting a 3 sigma process, you would only expect to have an RSD of 10%. So in those kinds of cases, you really have to consider that there are a number of different variabilities within your process, and you have to think about all of them. So in a lot of the cases for USP guidance, you may see that you can have a 5% RSD on your formulation concentration, and a 5%, and then you can have a 3 or 4% variability in terms of your filling process. But the thing that has to be remembered is that all of these variabilities, all these variances add. So what I show here, just as an aside, is essentially that you have to consider all of these variances, and together they have to be less than what is acceptable to the USP or the EP from that perspective. So all of these must be considered together, and not only are they considered together, but the way that we measure them, of course, creates variability as well. So that was just a little musing on in terms of how to think about variability when it comes to these types of processes, because there's a lot of different aspects to it. And then next slide, please. We just sum up in terms of what we've talked about here today. Combination product development requires integration of QBD design controls and human factors. Inhalation products have specific critical requirements, bioavailability and impaction being very, very prominent within that group. Manufacturers and laboratories can assist with the development for the processes, but the sponsor must be aware that ultimately, while we can assist with these things, the combination product development process must be controlled by the sponsor. So thank you, and we will take questions.
All right, thank you very much for that insightful presentation. So now we will move on to the Q&A portion of the webinar, and I've already received a few questions, so I will start with those. Our first question is dissolution testing required for inhaled drug products?
So I'll take that one. No, it's not required. It's used a lot in the context of bioequivalence, more so from a regulatory standpoint. But I think there's value to that in if you're developing a new product as well, and understanding, that dissolution profile, how it relates to the formulation, that microstructure we talked about. So there's growing acceptance of it. As I pointed out earlier, there's no real standard procedure, so that's probably one of the potential limitations. But I do think it's valuable, but it's not a regulatory requirement.
Yeah, I would like to add on a little bit, actually. I mean, I've been in situations where we actually ended up with a non-efficacious product because of the dissolution profile of the material, and while we could deliver to the right location, it wasn't able to solubilize within the in this case the nasal fluids that we're working with. So it's important, even if it's not required by by the FDA or by USB. You do have to think about it.
All right, thank you. Our second question, what is the target inhaled particle size?
So probably Justin and I can both touch on this. I'll start. Again, I think I'll be a little bit cheeky and say it does come back to understanding what your target profile is. What the disease indication is and where your those receptors are, or the target in the lung. But in general, particles that are less than 5 microns are considered to be respirable. So a 5 micron cutoff tends to be commonly used. However, if you are trying to deliver something to the deep lung or peripheral lung, then that parameter will be likely much smaller, different, like maybe 2 microns or 1 micron even.
Yeah. To add on that, you're absolutely right. It's important to, depending on where you're trying to deliver to. It also depends on what patient group you're talking about too. I think in certain cases, I think for infants and for children, there's different cutoffs in terms of what our expectations
Sure.
are. The other part is as you said, density is important in terms of factors as far as that goes. So we usually talk about aerodynamic particle size versus geometric particle size, and that's an important aspect as well in terms of how to consider this.
All right, thank you. Our next question, can you please share experiences on the level of device data for phase 1, phase 2, and phase 3 clinical material respectively, and to meet EU as well as FDA expectations?
Wow, that's a let's see, how are we going to answer that? Because the context of what we presented today is a little bit more aligned with the US, but the FDA, sorry, the EMA guidelines are fairly closely aligned. There are some additional considerations for European aspects, such as there's the 117 guidelines aspects, which we did not go in today. So I think that it might be a question that we could also help answer offline as well, because there are specific aspects really depending on the type of device and where you are in terms of what sort of information we would be providing throughout which would be provided throughout the process. Justin, I don't know if you want to add anything to that at all.
Yeah, there are a couple of things I think that are important, and then I'll probably be muddying the waters a little bit here, but when it comes to devices, then depending on the type of device, you could be talking about whether or not you're intending to CE market, as well as the biocompatibility aspects from ISO 10993, which also play a role, and even there can also be even 60601 aspects as well, which relate to electrical safety on such, depending on the type of device that you're talking about as well. I would say that what we did tend to do is to focus on the inputs to outputs, and then design verification aspects, and the human factors were ongoing in terms of this is previous experience and not where I am now at Experic, but in those kinds of aspects, we reserved only those human. We were considering the user requirements even early on in the process. We didn't validate any user requirements until very late in the clinical phase 3 stage.
All right, thank you. I believe we have time for one more question. It's a bit of a long question. I'm evaluating preservative-free multi-dose ophthalmic dispensers. Is there a requirement to show that individual dispenser materials do not swell with formulation contract, or is it acceptable to only show delivered dose uniformity and functionality of device over patient use?
I'll jump in, Justin. You can add to that. I think that I think the answer is yes from at minimum, you need to know this from a stability standpoint. If the dispensing materials, like if there's a gap, if there's swelling, or if the drug is absorbing into those materials, then you will have a delivered dose problem. So I think if you suspect that and your supplier cannot also help with an understanding of that, then I say the answer is that you should try to understand that as it relates to delivered dose. There's also a micro question part to this.
I'm
Yeah.
not sure I'm completely understanding either, but maybe to add to, we're talking about the container really in that
Yeah.
sense. If we are, then I mean, just from the biocompatibility and extractables and leachables aspect, you have to understand that and
Yeah.
consider. I mean, and
True.
it's both, and because it's a drug device combination, it's not just the ENL. You actually have to look at it from a biocompatibility perspective as well. So it's both in that sense. So it's, I mean, I think the answer has to be, I mean, very briefly, yes, but there's a lot of, obviously a lot of subtlety there.
All right, well, thank you very much for those answers, and we've reached the end of this Q&A portion of the webinar. But if we couldn't attend to your questions, the team at Experic may follow up with you, or if you have any further questions, you can direct them to the email addresses on your screen. Thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from Xtalks with access to the recorded archive for this event. The survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Justin Lacombe and Julie Suman. We hope you found this webinar informative. Have a great day, everyone.